Okay. Good morning, everyone. So now Professor Nirko will give last of his series of talks.
Yeah, so thank you. So thank you for staying until the last day. Only now I realize that it's on a Saturday morning, so you're very brave not to go away for the weekend. I'll use this opportunity to flash an advertisement on behalf of a colleague of mine, Anne, who is in Ben Gurion University in Israel, and she's an experimentalist who is making amazing in vitro actin and myosin gels. So she's using the basic components that build the cell cytoskeleton, the actin, the myosin, and other proteins that control the actin and the myosin. She puts them as they are, as these proteins are, in concentrations which are well controlled and with just a few components, unlike the thousands of components inside the cell. She's aiming to recreate the cytoskeletal dynamics in the test tube with just the minimal amount of components, number of components. And what you see here is an example of such an actomyosin gel, which he has created and is under the process of buckling and forming these three-dimensional pictures. The long vision for her is really to get something which will recreate some of the dynamics that I will show you in my talk now from the simulations, from the theory. So in some sense, this is an experiment going after theory, but it's a very courageous experiment, and she's looking for postdocs. So this was just my advertisement on her behalf. And if you're interested in contacting her, you can ask me or you can find her directly on the internet. So yesterday I showed you that this idea that you can have membrane proteins that are both curved and they induce active forces by recruiting the actin polymerization, the actin cytoskeleton, can initiate cellular protrusions. But now we will actually leave the limit of small deformations, which allowed us to do analytical work in the form of linear stability analysis, and now go into simulations of large deformations. So this is where we were last time with small deformations and linear stability analysis. What happens when you want to leave that regime and go into the regime of large deformations? So what I will show you is a collaboration that I have started already several years ago with Alesh and his wife Veronica and their group members in Ljubljana in Slovenia. And the first paper came out just a few years ago, and it is based on the notion that you can actually do a Monte Carlo simulation of a triangulated surface. So this is something which you already saw yesterday in Madan's talk, because Sunil is doing essentially a very, very similar type of calculation. The basic framework is identical, it's the same framework. And that allows us to calculate large deformations, just like Madan showed yesterday for his model of Golgi. Now this will allow us to look what happens if we have this coupling between curvature and forces, but not limited anymore to the small deformation regime. So as was introduced before, we have this bending energy, which we have to calculate for every node on this triangulated surface. So there's the mean curvature and there's a spontaneous curvature if you have something on the membrane. So for example, in this case, all the blue parts are bare membrane. They have zero spontaneous curvature, so this parameter is zero. And wherever you have red dots on the membrane, that would be your curved proteins. And you can even see with your eye that they tend to bend the membrane outwards, even just by their own shape, because they have a spontaneous curvature which is non-zero. And in addition to that, they also tend to bind with each other. So there's some binding energy W, which is only working for nearest neighbors. So it tends to connect nearest neighbors with some binding energy, and that's why they aggregate. So that's another energy term that you have to include. And what we want to include, which is the new term, which was not there before, is the active forces. Now, the simplest way that we could incorporate the active forces into this Monte Carlo scheme is by adding an energy term. So we have to convert this force into an energy term because the Monte Carlo is calculating only energies. So what we did is the following energy term. So if it's sort of represented schematically by these arrows. What it essentially means is that if you have a point on which the force is exerted, which is only the places where you have these proteins, and you displace this node in the direction of the outwards normal, in the local outwards normal direction, so this dot product is positive, then you lower the energy by an amount which is determined by this prefactor minus F. And F is indeed units of force, and this is units of distance, so together it gives you, in essence, the work done by this force in moving this point in the direction of the force. So it's as if there is an external field acting on each of these points, pulling it outwards, okay? Now this is, you can say, okay, but why would you call this an active force? After all, you are just simply adding it into the energy term, and you don't call these active terms. So what's active about it? What's active about it is that unlike these energy terms, which are bound from below, there is a really absolute minimum, global minimum to these terms. There is no global minimum to this term. It can go off to minus infinity. So if I grab each of these points and pull it and pull it and pull it to infinity, then I'm just going down an energy slope which never ends. So in that sense, it's not a legitimate energy term that I can really minimize. It's really a representation of an external field, and an external field means that this system is not closed and I'm not really conserving energy, or in other words, I'm pumping energy into the system. Okay, so the membrane term you saw before, it has this spontaneous curvature, only where you have a node which has 1 for this row, which denotes that there is this inclusion or there is this protein, and for the bare membrane, this parameter is 0, so there's no spontaneous curvature. There's the binding between nearest neighbors, so if you have a nearest neighbor, then you have a minus energy of W for the nodes that are occupied by these proteins, and of course there is 0 binding between nodes which have no proteins. And of course there is this active force, just like we said now, which is pushing outwards on every point where there is a protein, and it's to take into account this acting polymerization, which we don't take into account explicitly. So there are simulations that you can find in the literature where they do a much more complicated calculation, where they are trying to simulate the individual filaments and how they're really polymerizing, depolymerizing, branching. It's a much more heroic effort, but we avoid all of that by simply saying that all of this structure is simply just giving us an external force.
A question?
Yeah.
Here. So the membrane, does it have also it has a bending rigidity, right?
Yep.
And there is no spontaneous curvature in locations where you don't have the protein sitting. Okay. So in the absence of protein, this will be flat.
Well, it's closed into a sphere, so it will be a sphere. Yeah.
But how the sphere is maintained?
Because there is a fixed area.
Okay.
So you want to minimize the curvature energy for a fixed area, so you just make it into a sphere.
Okay.
Okay. Otherwise, if you want to have parts which are flat and super high curvature at the rim, that will cost you much more. So how do we simulate that? I will not go into too much of the details, but in essence, you roll the dice and you get you see if the next move, the next Monte Carlo move, is accepted or not accepted by the Boltzmann factor probability. So, for example, the initial conditions can be something that looks like this strange tetrahedra, and then very quickly you see that it assumes a form that minimizes the overall energy. There's also thermal fluctuations here because there is a finite temperature. So each Monte Carlo move is accepted or not accepted by the Boltzmann factor with the temperature. Of course, this is a very low resolution simulation with very few nodes, just to illustrate the way it goes. As was mentioned by Madan yesterday, there are these node flipping events which allow us to actually have diffusion in this membrane. So that's just a summary again of all these energy terms. But yeah, there should be here the term showing the bond flipping term that allows you to actually have bonds changing from one topology to another topology, and that allows each of these nodes to diffuse on the membrane. So the membrane is maintained as a fluid. Although it looks to you like a triangular solid, we constantly flip the topology locally with these Monte Carlo moves so that each node can actually diffuse around. So it is a fluid, and we can show that things are diffusing around it. Okay, so if you have this system and you begin to simulate, and you have these proteins which are curved outwards, and it is a passive system, so you have no forces. And you only ask as function of temperature, for example, which is the y-axis, and density of these proteins on the x-axis, what kind of shapes do you get? So, and in each inset, what you see is that you see the size distribution of aggregates of these proteins. So when the temperature is high and the density is low, you basically have a very dilute gas of these curved proteins on the membrane, and you see that the size distribution of these aggregates is peaked at 1. So basically you have many individual ones and few pairs, even fewer triplets, and so on and so forth. As you increase the density, you see that the whole vesicle begins to sort of have funny shapes, because more and more of the area is covered by these proteins which are highly curved, and they begin to aggregate, but still you don't see anything very dramatic about the size distribution, although there are now more and more larger aggregates which you can pick up also with your eye, and because they are so curved, they curve also all the membrane around them. If you lower the temperature and again increase the density, all this budding and aggregation activity happens at a much lower density. And what is shown here by this straight line is in fact the calculation that you can do from the same linear stability analysis that I showed you yesterday, and that gives you what is the critical temperature as function of density for the system to become unstable, which is a little bit like calculating the spinodal of the system. When does it begin to want to phase separate into aggregates? And it fits rather well with the contour that you get from the simulations for the mean cluster size of 2. It's not very impressive to have a cluster of just 2 proteins, but if you take that as sort of a contour line which divides where you are below pairs, so you're really in a gas phase, or above pairs, so you begin to talk about budding, it fits rather well with this linear stability analysis. You can do a more involved calculation to try to pick up the line of when should you begin to find these highly paired structures, where in fact many of these buds link on top of each other in order to minimize the energy. I'll show you a close-up on these shapes in a second. Okay, so first of all, that's the linear stability analysis result, which gives you this straight line. And you see that it fits rather well with this contour line from the simulation of mean cluster size 2. So it just shows you that linear stability analysis is still useful, even in a situation where eventually you'll be interested in highly nonlinear shapes like these ones. Yeah, so what happens when you get these shapes? Why do you get these shapes? So you get these shapes because it minimizes the energy, because if you think about it, each of these buds, they want to form like small spherical objects. I didn't actually say that, but it's a good opportunity to say it now, that these curved proteins are isotropic. So we took them to be such that they're really parts of spheres. They're not banana-shaped, for example, which would mean that they would be perfectly fitting along a cylinder by having high curvature in this direction and perfectly straight in the other direction, or even saddle shape, or all kinds of other anisotropic shapes that you can think about and that do exist in the cells. We made our life simple by just taking an isotropic shape. So it's perfectly part of a sphere, which means that each of these proteins, if it's rotated in the plane of the membrane, it doesn't matter because you're rotating something which is circular. So they themselves, they just want to make small spherical buds, but wherever they form such buds, there is also a contact line with the rest of the membrane. So by building themselves on top of each other, they minimize the contact line with the membrane, which means they minimize the places where red proteins don't have any more red proteins to bind with. So they're minimizing the loss of this binding energy, right? And you can see that you can do a semi-analytic calculation. It's also numerical, but it's not a real simulation. It's a minimization of the energy for the situation where it's perfectly cylindrically symmetric. You must do something to simplify, otherwise you cannot solve it, and you get similar phase separation and similar shapes. So that's still all in the passive case, okay? So we haven't turned on yet the activity, which will be very dramatic, as you will see. When I show this image to somebody, then he sent me to this very old paper by Helfrich himself, from the Helfrich energy that you've heard so much about, and you can see something which might be even similar, but again, it might not be related. Such pearl structures, they call these pearl structures because they look like pearls on a string that you can wear as jewelry, and you can find many pearl structures in experiments whenever you have a membrane that has some form of curved proteins on it. And not much more beyond that. In this passive system, as I said, as function of temperature and density, you can plot the mean cluster size, and of course the cluster size is increasing as temperature is decreasing and density is increasing. That's the contour line of mean cluster size 2. So you can roughly say that you have a mixed phase here and a budded phase here. And you can see that the linear stability analysis is working very well because, for example, if you plot the mean cluster size as function of temperature for two values of this binding between the proteins, you can collapse all of the data from the two sets of simulations if you simply rescale the temperature by this critical temperature that you calculated from the linear stability analysis. And the same goes by changing different densities and again calculating the mean cluster size, and everything collapses with the single energy scale which is given by the same linear stability analysis scale. So you see that linear stability analysis is not useless. It can actually give you a good handle on what goes on, even in systems that eventually become highly nonlinear. Now we turn on activity. So when you turn on activity, this is on purpose missing. At first, when you have high temperature again and low density again, not much looks very different. What is different is that you can actually calculate that this linear stability analysis line is shifted. And this shift you can actually calculate. I hope it's shown here. So that was the line that what you calculated for zero force. Now that the force is not zero, it's shifted to higher temperature and lower density. And you remember that was our sort of qualitative explanation of what happens for the HIV budding, that the virus is recruiting the actin and is therefore able to shift its budding transition from high density to lower densities and begin to bud out from the infected cell at a lower areal density of the membrane. So this shift was what we saw before, and you can see it here. And again, if you plot the calculated contour of mean cluster size 2, it seems to be shifted similarly compared to what it was before you turned on the force. So at least, again, for the limit of just small buddings, again, linear stability seems to work. But here, at the low temperature regime, we find a completely new phase, which took us by surprise, and it's sort of the line that marks the appearance of this phase is denoted by this red dashed line. It's roughly. And you can see, from the insets that instead of having a distribution of sizes of clusters, okay, here already you can see that there is some bumps, so that there's some typical clusters of larger size, which are these pearl structures. But here, all the proteins are really like a delta function at a single huge aggregate which essentially holds all of the proteins together. So they spontaneously phase separated, perfectly into one huge aggregate that is now all around this object. So if you cannot actually make what this object looks like, there will soon be a zooming in. What is interesting is that on this side of the transition, at low densities, this is a very sharp transition, as you can see here by zooming in on it. Below a critical density, or basically a critical number of these proteins per this fixed area of vesicle, of membrane, this pancake shape cannot be sustained anymore, and there's a sudden shift to another class of shapes, which I will show you in more detail how it looks like, but it's highly elongated and looks very, very different from these pancake shapes. And it's simply due to the fact that there's not enough of these proteins to fill a perfect circle around this pancake shape. And once you cannot fill a perfect circle, there is a gap, then it means that the forces are not anymore perfectly balanced and the whole system breaks apart. So we'll talk about that in a second. So what's happening here? Why do we get this pancake shape? So in the linear limit of small fluctuations, you remember that the force was actually helping the aggregation. And that was indeed why this linear stability analysis line for the budding was shifted to lower densities and higher temperatures. And that was simply because each of these proteins wants to curve outwards, and by having the force also pointing outwards, it's sort of making each protein able to deform even more than it is able just by its shape, and therefore this aggregation tendency of them to go together in the place where more of them are deforming the membrane in the direction which fits their curvature is enhanced. So this is very intuitive for the limit of small aggregation and small deformations. But when you have lots of them in one place, you remember that they want to form these perfectly spherical buds. But now, since the force is perfectly normal in all directions, it actually destabilizes such small structures. So you can understand that the same force that was actually helping to initiate the aggregate is also now breaking up the type of aggregates that these proteins want to make just because of their shape, which is a perfect small sphere. So the next best thing that the system can do is actually to have all of these proteins on the rim of this pancake, because then if you look at the forces, all the forces are in fact radial with respect to this shape, so they're all stretching the membrane in the same direction, so they're all helping each other now, because they're all helping to maintain this stretched shape. And you can see that it is very, very stable. There's hardly any event that you can recognize where a protein leaves this structure and begins to meander in these flat regions, although it can do that. And as soon as this structure is broken, as you'll see in the next movies, you'll see that they readily break up these aggregates. The binding is not so strong. The binding is taken here to be something like 1 kT. So it's not in a regime where binding is so strong that they cannot unbind. In fact, if the binding would be so strong, it would kill this pancake shape, because it will make the proteins do what they really want to do from their own shape, make spherical buds. And then these spherical buds will be strong enough to resist the force, and you will get only back to a shape where you have a spherical free membrane and budded proteins, because the binding between them is so much stronger than the active force that they don't care about the active force. They just want to do what they want to minimize the binding energy. But because the binding energy is not so strong here, they are able to make this pancake shape, but still the proteins find it highly undesirable energetically to live and meander here. So they are very happy from the high curvature that they are now all maintaining for each other, at least in one direction, but they are not happy with the curvature in the other direction, right? Because in one direction, this is really very thin, and they really have the high curvature that they want, but in the other direction, the radius of curvature of such a big pancake is very large, so it's very far from the curvature that they want, and indeed, That's the reason why, if you look at the edge, it is constantly sort of frustrated by that, and it's trying to bend also in this direction, but bending in this direction and making this into something that looks like a spherical bud, which is what these proteins want from their own shape, is now not sustainable. It's not stable, and then these petaling shapes will immediately straighten and petal again and straighten, and that's why you have these constant fluctuations at the rim. So these fluctuations are there all the time because of this frustration of the curvature of these proteins. They're happy in one direction, but they're unhappy in the other direction. But it still doesn't destabilize the whole structure. Now, of course, you know, when you look at this, you say, oh, okay, that's, Nir is telling us about it already in 5 minutes. We understand everything. And it's so intuitive. But apparently, none of us who did this simulation, this study, were thinking, yes, that's what we expect to find. Nobody was saying that, which I think is a lesson for you when you're running simulations and you are allowing the system to have large deformations, large nonlinearities, that it's very difficult for you to envision what will happen. Although this system is really very simple, only membrane, curved proteins, and forces which are always directed normally outwards. What can be very difficult for us to envision what will happen? And still, we're all the time surprised by what we find in these simulations. Only in retrospect, you say, oh, yeah, it makes perfect sense that this should be stabilized by this system. But as I said, we didn't know it before. Okay, if we zoom in again on this region of low number of these proteins, so now there's just not simply enough of them to complete a full ring. So there cannot be a perfect force balance around the entire circular rim of this structure. So what happens? So what happens is that they are trying to do something as similar as possible, and what they end up doing is that they end up making two arc-like aggregates. So they make an arc-like aggregate here and an arc-like aggregate here, which maintains something which is rather flattened at the two ends, but in between you have a membrane which is basically now pulled apart because the net force of this arc is upwards and the net force of this arc is downwards, and the membrane in between is basically pulled. And you know that if you pull a membrane, you get a membrane tether, or basically a cylinder. And that's what you have here. And you can pick up with your eye immediately that there are many isolated proteins now meandering around, which tells you that it's now much easier for these proteins to break up from these arcs and move and join the arc and leave again. So everything is much less stable as far as these aggregates are compared to when you slightly increase the number and you allow them to make this perfect pancake. So again, you can sort of understand what goes on here, at least intuitively. Now, could this mechanism be related to these flat protrusions that cells are making when they are moving on the surface or inside some gel, which is called lamellipodia. So we mentioned this already without giving it this name many times, that we said that when a cell is moving on the surface, it is, so initially it spreads, there's the nucleus here, and then at some point it breaks symmetry and it makes a protrusion, which, and there is now a round back, and the whole cell is moving, let's say, to the right here, and this is full of this actin which is pushing, and this has the name of lamellipodia, and it is usually very thin. Now you can say, okay, it might be very thin because of the adhesion, and here you have no adhesion, this is sort of floating in space. So could that be related to this? We'll see. I hope to convince you in this talk that we think it does. So if you look at movies of cells that are moving through a gel, this is a very beautiful light sheet movie. You can see that the cells spontaneously form these sheet-like protrusions, which you can call them lamellipodia, you can call them ruffles, but they are certainly very flat-like. And the question is, of course, what makes them, and why are they so flat? And here I'm showing you, of course, a simulation of this model, and the idea is to suggest that this is actually flat because there are these highly curved proteins at the tip of these structures, which once they are produced by the cell and they can induce actin polymerization, they spontaneously form these flat protrusions. That's the new idea. And do we have any evidence that such proteins of this kind of curvature exist at the edge of the lamellipodia? So there are several recent papers. This is 2019, this is 2021. This more recent one actually suggests that it's even saddle shape. And that, again, as I mentioned before, should point out the way to actually describing proteins which are not isotropic of the kind that we have done. So the isotropic proteins that we have done so far, and that all I will show you today, are just simpler. But it's always good to start with a simpler one and understand what does that give you, which you'll see is already a lot of results before you move to the more complicated proteins, which seem to be there at the leading edge of this lamellipodia. This, what is called wave complex, is exactly a protein which is sitting at the edge and is inducing the actin polymerization. So again, it is presumably doing what we want from this protein. Now you can ask, is activity alone giving rise to this flat protrusion? So is it really the combination of curvature and activity, or would I get it also if there would be just simply pushing out? What do you think? Okay, you don't want to guess, so because I scared you before that I told you that it's difficult to envision what will happen in these simulations, so it's my fault. But we also didn't guess, so that's why we did the simulation. So first of all, again, this is blocked on purpose. When you have high temperature, again, you have just aggregates, and you can already see that even though you have quite big aggregates that you can even pick up with your eye, you don't see any budding out because these proteins are now perfectly flat. They're just as flat as the blue parts. So the red and blue parts are just equally flat, only that the red are pushing out. Okay, but they're pushing out with such a force that here at this high temperature, because the aggregates are small, you don't really see that they're pushing out. I don't know, maybe this is slightly deformed, but you know, not much beyond that. But once the temperature is low enough so that simply the aggregates are big enough, because the size of the aggregate is always controlled in this case by temperature, entropy versus binding, you get these shapes. So these are very different. And again, I guess you couldn't have imagined that, but these are not the flat structures that we saw before. So you will agree with me that in order to get flat protrusions, you must have the combination of curvature and pushing force. Pushing force by itself is not enough. And in fact, only when you look at this again, in retrospect, you say, aha, of course, a flat protein with force should give me a protrusion. A tether-like protrusion, a cylindrical protrusion. We call these hydra shapes because they look like hydra in some sense. And in fact, they're also very dynamic, unlike this flat shape, which was very, very stable, except for these small fluctuations around the rim. These are constantly changing. So the length of each of these protrusions is constantly changing. These aggregates that you see clearly at the tip of each of these protrusions are sometimes merging with each other, breaking again. So already you see here what is the answer. The answer is that you get an aggregate of these proteins. As you remember, each of these proteins is giving a pushing force. So simply when you have an aggregate which is large enough, it's able to pull a membrane tether. So that's all there is to it. And you can actually do this calculation. Do I have it here? Yeah, okay. So before that, I just want to show you that indeed, as function of the curvature of these proteins, no, this is, sorry. Yeah, so So yeah, so you see that first of all, there is, so that's not a function of curvature. As a function of the temperature, you can see clearly that you transition from small aggregates that are not big enough to form the tethers to aggregates that are large enough in order to form the tethers. There's a clear transition. And you can ask yourself again, can we do some calculation here which will explain to us what's going on? So indeed, you can do this force balance calculation, which I think by now it's easy for you. So the pushing force is given by this size of this aggregate, which is basically the size divided by the size of a single protein. So it's the number of proteins in this aggregate times the force that each one of them is producing. And let's assume that we ignore that it's slightly hemispherical and just treat it as perfectly flat so that all the forces add up in the same direction. And what is the restoring force? So the restoring force comes from bending the membrane. So the bending of a tube gives you a force, which is the energy per unit length of the bending of a cylinder, of the bending modulus times 2 pi over r. So by equating these forces, you can get a relation for the critical radius at which these forces balance and this is strong enough to equal this and to begin to draw out a membrane tether to be related like this. So the relation between the radius or the size of this cluster to the force and to the bending modulus goes like this power of 1/3. And then you can go and do some simulations, and it's, roughly in agreement, I would say, with this third power, 1/3 power, between this radius of these clusters and this combination of kappa over F. Now you can indeed see that as you change the spontaneous curvature of these proteins, when they are highly curved, they form these flat structures, but then at some point, they cannot anymore sustain this curved structure because of they become too flat, and there is a rather sudden transition into a different class of shapes. One of them is more ill-defined. You can call it, we called it like a puddle because it's elongated, but it's still got flat parts at the end. And then you have these, what we call the hydra shapes, which are only made out of these cylindrical tethers. Yeah, now you can ask, what about adhesion? So a very good postdoc of mine, who is now continued for another postdoc, a second postdoc in Institut Curie in Paris, Raj, did this work, and basically he added adhesion to this problem. And one paper already was published last year, and we have several papers now in referring. And the idea is to indeed ask what happens when adhesion enters the story, because you can say that, yes, you got these flat protrusions even without adhesion, just in floating space, but clearly adhesion will affect things because adhesion might also want to get things into a flat shape just by adhesion. So what will be the interplay between these two things? And specifically, could we get something that is really polarized in one direction and moving, which, this flat pancake was nice, but this pancake was just floating in space, was not really moving anywhere. Okay. Yeah. So you need another energy term, and the other energy term that Raj added is very simple. He basically said, I mean, you can think of more complicated things, but again, let's start simple. Any node of the membrane, doesn't matter if it's a membrane node or what we call these protein nodes, the red nodes versus the blue nodes, all of them, if they are within a distance of delta z from the substrate, which is just a geometric plane in space, you have some additional adhesion energy minus EAD per node, or per unit area represented by a node. Okay? So it's the simplest thing you can think of. It's really like a step function. You can think of more complicated things that might emulate some springs, but This was the simplest thing one could think of. It might mean, as you know, remember this simplification, because it might mean that when you're comparing eventually your simulations to real experimental data coming from your friends that are doing experiments, it's not enough. You have to add a little bit more complexity, but it's good to start simple, but be aware that you might need to add more complexity down the line if you want to compare to experiments. So first of all, again, start simple. Let's start with a membrane that has no proteins. Yes, go ahead. Yes, all the nodes which are in contact, they add this minus energy term. Yeah, you run the Monte Carlo simulation, and any node which enters this small distance with the surface now gains this minus EAD energy. Any node that leaves this region loses minus EAD of energy, and that tells you what will be the probability of accepting or rejecting that Monte Carlo move because of the Boltzmann factor of the energy. Exactly. Okay, so this is proteins, this is vesicles without proteins, just to show you that we can do adhesion. You see that the adhesion energy here is very low. Can you see no, you cannot. So this adhesion energy here is very low, and you can see that it hardly spreads on the surface. It doesn't leave the surface, but it hardly stays on the surface. If you crank up the adhesion energy to 5, then you see that it will spread much more. What is preventing it from spreading more and more and more? What can be the balance of energy that prevents it from spreading? Because by spreading, it gains this contact area, which is lowering the energy. So why doesn't it continue to spread more and more? Not really. There is no real surface tension in the sense that we are preserving all the time the area. So area doesn't really change. There's no active forces here. No active force, no proteins. So there is high energy because there is high energy cost to pay by bending the membrane, because the membrane has this rim around which is highly bent, and that costs a lot of bending energy. So at some point, it stops at different values of spreading at a point where this energy from adhesion is balanced by the cost of bending. So you can see what I mean by looking at the cross-section. The more the vesicle is spread, there is a higher cost of bending energy around the rim. And what is compared here is the simulation to a numerical solution of this problem under the conditions of perfect cylindrical symmetry that was published by Jill and her supervisor, and it sort of gives us just a sanity check to show that our simulations are not so bad. They are not perfect, but they are not so bad. You can get all kinds of very funny shapes when you have the solutions of a membrane with different ratios of volume to area and different strengths of adhesion. So if you're interested, you can read all about these analytic solutions or semi-analytic solutions in this paper. But it also gives you an opportunity to see that we don't conserve the volume. Now we can add something which is similar to volume conservation by adding osmotic pressure. So different strengths of osmotic pressure will tend to preserve more and more the original volume until the pressure is so strong that basically it kills all the shape deformations. But all I will show you has zero osmotic pressure, and therefore the volume is not conserved. Now we add these curved proteins, but again, we go one step at a time. They are passive. So they're just curved proteins. They like to aggregate. You saw they would want to make these purled structures when they are just on the free vesicle. But now there is adhesion. So what happens? You can see in this sort of phase diagram, if you want, as function of the adhesion energy and density of these proteins. So with low adhesion energy and low density of them, you have very weak adhesion. Now what happens if you keep the adhesion strength of the surface itself, of EAD, and you just increase the number of these proteins on the vesicle? You see very strongly that there is much stronger spreading. And you can immediately pick up with your eye why this is actually spreading so much more, because these proteins are these curved proteins are all aggregated on this highly curved rim. And they like this high curvature. So instead of paying energy in order to bend the membrane for this highly curved rim, you're actually now enjoying this highly curved rim by aggregating these proteins exactly where there is the high curvature that they like. And you see that, of course, if you add too many of them, they need to be somewhere. They begin to form these pearl structures again, which just shoot out from these rims. So you can see this in these two movies. So we on purpose choose a very low adhesion energy. So without the proteins, there is minimal adhesion. And as soon as you add quite a lot of them, 17%, you will see that it spontaneously spreads very nicely on the surface just by virtue of segregating these curved proteins on a rim spontaneously because they like this high curvature. And you can see it in this graph here, which I think summarizes it very nicely, that if you look at the bending energy as a function of how much of the area of the vesicle is adhered. So if you have no proteins, the only way to increase the adhesion and the adhered area is to increase the adhesion energy. But as you're increasing the adhesion energy and you are spreading more and more the vesicle, you are paying more and more bending energy of the membrane because of this highly curved rim. So you can increase your spreading, but at the cost of increasing your bending energy. And in order to offset that, you have to increase the EAD. But if you fix the EAD, you don't touch the adhesiveness of the substrate, but you just increase the density of these curved proteins, you can also increase your spreading. But now, as you're increasing your spreading, you're actually decreasing the overall cost of bending energy just by the fact that you're actually able to aggregate them spontaneously around the rim. And you can see that if you look at the angular distribution of them, it's highly spread everywhere before it begins to spread. But as it is fully spread, you can see that they are highly peaked along the rim. I mean, you can pick it with your eye, but you can also quantify it. Okay, so let me jump over these movies. And now we turn on the force. So now you turn on the force, you look at what we call this phase diagram that we saw before as function of adhesion energy and density. Now again, at a high density, you find these highly spread objects with vesicles with all the proteins on the rim. So you can say, okay, that's very similar to what I got without the force. On the other hand, it's also very similar to what I got with the force without adhesion. When this thing was floating in space, it also looked like that. So I'm not sure what really I'm seeing here. It can be both from just simply the force making a pancake, also in space, just now shifted onto the surface, or it can be something different. And you can see that if you don't have enough of them to form the perfect pancake, again, you make these two arc shapes with a highly elongated middle part. So it's already clear that this is very different from the shapes that you got without the force, which was just simply a cell which was almost not touching the surface. And what you can already see from these movies, and also from this phase diagram, if you look more carefully, is that the big advantage that you see so far from putting on the force is that you can actually be in a regime where the passive case doesn't actually spread out very much because the density is just not enough. The density, in order to spread very well with the passive proteins, needed to be very high, almost 20%. So as soon as you go to 10%, this effect is not very significant anymore. You see that there is some spreading, but it is not very efficient. And with the same low density of 10%, once the force is turned on, you can see that you form these very nicely spread out objects. So immediately from this, you can say, aha, by turning on the force, the cell can actually spread on a weakly adhered surface with a much lower number or much lower amount of these curved proteins that it would have needed to create if it would be just using them for their passive property. So that's one advantage for the cell. And indeed, as you go to much lower density, like 3.5%, you begin to form these two arc shapes because you just simply don't have enough to produce this perfect pancake. So you produce these shapes which are highly elongated and the two ends of them have these arcs on them. Okay. Yeah, so we tried a simple analytical model to include the force. Yeah, I wouldn't say that it was so great, because for an analytical model, you need somehow to parameterize the shape, and the shape is not easy to parameterize. You see already that there is one region which you parameterize with one radius, and then there's a cap part which you parameterize with another radius, and there's of course the part which you are parameterizing, which is the contact part, the flat part. It's an involved analytical model. You work very hard, and at the end of the day, you only get qualitative understanding, which might be useful, but it's very far from being quantitative. It again shows you that even for these simple systems, as soon as the deformations are large, it is very difficult to do something analytic. Okay, you can actually look at the spreading dynamics, and you can also compare the spreading dynamics in the case where there's no proteins, the case where there is these passive proteins. The dynamics looks rather similar from the point of view of the growth of the adhesion fraction of the membrane area as function of time. But as soon as you turn on activity, you can actually have very strange shapes. For example, you can have these parts which seem like to go upwards before they go on the surface. It is shown here in this movie, I think. So you can get a much more dynamic state, which is sort of fluctuating upwards before it actually settles down. And this might seem completely crazy, but in fact, when you look at cells, they do that. Their edges are not simply spreading like a drop of water, especially when the cell is alive. It sometimes makes these large motion upwards before it settles on the substrate. So again, it shows that we can get some of these dynamics as well. I think it is even shown here in these snapshots where you see the edge of a spreading cell, and many times the edge is sort of lifted up before coming down again. But the biggest surprise, again, so far you said, okay, so we got the pancake and the two arcs, but we saw them also when we were floating in space. Now they're simply spread on the surface, so there is an additional adhesion energy. Not a big surprise, but then the surprise came. The surprise came because in the same regime where you have very few of them so that you cannot make a pancake, we saw these two arc shapes. But apparently, if you go to a different regime from where we were, which was low adhesion energy and high force, you actually reduce the force and increase the adhesion energy, you can get into this regime where the color code shows you the probability of finding another shape. And this other shape has all of the proteins in a single arc on one side. So if all of the proteins are on a single arc on one side, the force is not balanced anymore like in the case of the two arcs, and the system should be actually moving. And that's what will be shown here in this movie, where you actually have now a crawling vesicle or a crawling membrane, which is very similar to this polarized cell that we were aiming at or hoping that we will get, because these cells that are all in this case, this one's moving upwards, are very different from the perfect pancake cell on the surface by having all the actin polymerization happening on one side. Actin is labeled here in bright color. And this is another example of another cell. Again, all the actin polymerization is happening here, and it is moving in this direction. So we got a polarized object, which is really moving. And again, you can ask why. Why would this happen? So what you see if you look at the cross-section is that the front is very, very sharp, which has this high curvature that these proteins like. They don't exist in the back, so the back simply tries to minimize bending energy, so it becomes like a fat object. And we can at least analytically understand why there is this transition between this two-arc shape and this crescent shape, and that analytical calculation gave us this straight line here, which at least gives not such a bad estimate for the upper bound of this region where the crescent can actually exist. And I can show you this kind of approximation. Again, just like in the case of the hydra that we saw before, it's good to try to do some analytical calculation to give you some understanding. And as you see in this case, at least it works quite well. So the kind of analytical calculation is as follows. We try to compare the energy, including the work done by the forces for maintaining the two-arc shape. So for the two-arc shape, when we want to compare the energy difference between the two-arc shape and the crescent shape. So what terms do we have here? We have the energy which is doing the work which is done by lengthening this compact object into this two-arc shape when it will transition from this to this. So there is this additional work which is done, which is NF, so the number of proteins times the force that each one of them is producing times the length, that's the total work. Then there's additional difference because of this bending of, so we took something which had very low bending energy because it was almost like something which was not highly bent. And then, I mean, at least from the membrane point of view, the membrane here is not highly bent behind the proteins, and then suddenly it has to go into this very thin cylinder, so it pays the bending energy of the cylinder. And we also lose some adhesion energy because you can see that this crescent shape is much more well spread on the substrate compared to this object, which has some adhesion, good adhesion here and here, but all along the middle line, there is very minimal adhesion because you have a cylinder which is only touching the surface at a single line, at a single point. You can almost ignore it. But we don't know exactly what is this area difference. This is difficult to calculate. So unlike all the others, so when we do this estimate and we find numerically that this area difference between the two shapes is rather constant, there's no big difference as you go along this transition line, because we can treat this delta A as roughly a constant, we can actually calculate the relation between the adhesion energy and the force, which will give us this critical line, because we can treat this area difference as rather constant. And that gives us this solid yellow line without any fitting parameters, except for this value of delta A that we have to grab from the simulations, and it gives us a good understanding of what goes on here. So it's really a balance of energies which flips you from preferring the two-arc versus preferring the crescent shape. Now, it also gave us an opportunity to compare to experiments. For example, what do you see here? You see that, in fact, if the force is reduced, an object which was not motile, was not polarized, it was in the two-arc shape, actually becomes more crescent-like and more motile and more polarized as you reduce the force, which is a little bit counterintuitive, because in the case of cells, that would be like reducing the amount of actin polymerization and actually increasing the likelihood that the cell will begin to move. It sounds like strange. But in fact, we talked to Rob Insall and this group, and they sent us to this paper of theirs where they took cells which are not very motile. You see that they have many leading edges, many of such arc-like objects, if you want, which prevents them from polarizing in the normal case, in the normal type. And then they reduce some of the proteins that, or knock out some of the proteins that polymerize actin, so the cell is left with less actin polymerization. And you can see quite clearly that it becomes very crescent and polarized, very similar to this transition that we envision here. So again, you reduce the amount of actin polymerization and you actually get a polarized state. Now, after we were so happy to get a polarized state and a moving cell, or something that looks like a moving cell, you also have to remember this, the victory is not all yours yet, because this system is indeed super, super simple, and it manifests in being super fragile. For example, you can see these snapshots, and I'll show you the movie that goes with these snapshots in a second. As time goes by, by chance fluctuation, there was like a nick, like a small break in this large aggregate of these proteins. As soon as there is this nick here, this break here, the net forces from one part of the aggregate are slightly shifted upwards, and the arc here is slightly shifted downwards, and now there is no way back. They are slightly increasing and enlarging this break until they end up in this two-arc shape, and that's it. I mean, the chance for a chance fluctuation that will bring this thing back to here is almost negligible. And you can see it actually happening in the movie. So it doesn't look like it's moving because the camera is sort of glued to the center of mass. And then at some point, there will be this fluctuation, and then very rapidly, you will see that the two sides basically break apart. So it's a very fragile system, and you can say, okay, that means that it cannot be in cells, but in fact, in cells, you do see many occurrences where the leading edge of the cell, so this is a cell moving to the right, and this is the leading edge, which is full of this actin, it can actually form a break and disintegrate into two opposing leading edges, but the cell has an internal mechanism of repolarizing. And you can see that one leading edge was winning over the other, and the cell just left off and continued walking in a new direction. And we don't have a mechanism for that yet, and that's one project for a new postdoc that I have at the moment, Shubhadeep, who's working on that. And you can see another example of that from this movie that comes from Mathieu Copé in Curie, where, again, a cell that looked like very well polarized at some point spontaneously disintegrated into this two-arc form. But again, it has a way of resolving this shape. One of the sides detaches and loses its actin polymerization, and the other one wins, and the cell repolarizes. And we don't have that. But this transition by itself does happen in cells. So it's not like that as soon as we see this fragility of our polarized state that it means that it has nothing to do with reality. Now, you might have also asked, but you were too shy, that the two arc states that I showed you before, I said that they have force balance, but why would the two arcs be always perfectly the same? There's no reason for that. And indeed, you can get arcs that are not perfectly the same. And if they're not perfectly the same, there is some net imbalance in the force that will lead to some crawling in some direction. And again, you can see that sometimes in cells. And again, in cells, this is not very stable because cells have an internal mechanism for resolving this and eliminating this small competitor and making everything go into the main leading edge, which we don't have at the moment. Another demonstration of the fragility of this system is what happens when it hits a wall. So when it hits a wall, because it hits a rigid barrier which is perfectly flat, it immediately destroys this high curvature of this part which is hitting the corner, and then all these curved proteins need to escape from there because they don't like to be flat. They want to be in a curved place, and they end up shifting into two arcs, and that's it. As far as our simulation is concerned, it will never recover. But again, cells do undergo such loss of polarization when they hit a barrier, but they have a method and internal mechanisms of recovering polarity and moving off the barrier, which we don't. Okay, you can also consider more smooth barriers, such as this sinusoidal surface, etc. Now you can ask the following question, which we wanted to do. I have some time? Yeah, I have a few minutes. What happens if you now let this same vesicle that we talked all about now adhere to a round object? So let me up. No, not good, not good. Okay. So you can see that we chose a binding energy which is very, very weak. Don't go. This is for surprise at the end. Okay. So you can see that it is very minimal. So we chose on purpose a binding energy and adhesiveness of the surface which is very weak. So without the proteins, there is hardly any engulfment of the vesicle of the bead by the vesicle. By the way, phagocytosis is the process by which cells engulf objects that are outside of the cells. It could be by immune cells that eat up bacteria or other garbage in the body that needs to be cleared out. So it's a very, very essential process in biology. And it's true that you can think of it as just a problem of adhesion, a membrane that adheres to an external surface. In this case, it's a perfect bead, but a perfect sphere, but it can be something elongated like a small bacteria. But it is known that actin polymerization is essential for this process. So the cells do not rely only on just a super strong adhesion. In fact, there is never a super strong adhesion in nature because you always need to move off. So if you would have a super glue, then it's not going to do you any good when you start to move away. So you want to be able to spread very nicely on weakly adhered surfaces where the basic adhesion strength per unit area is not large in terms of KT. Now, when you add those passive proteins, passive curved proteins, you remember on the flat surface, they did help us a little bit or even a lot if we had enough of them to engulf or to spread. And you can see already with your eye that the spreading is slightly increased, but we just didn't add enough of them. We could have added a lot, and that would have completely engulfed by the same mechanism that you saw on the flat surface, just by relieving this high bending energy that this vesicle has to pay when it tries to begin to engulf. It needs to bend over these edges as it is trying to do this engulfment. And again, if you add many, many of these passive curved proteins, that can result in full engulfment in this situation. But on purpose, we put here just a few of them, so it's not enough. But with the same number, just a few of them, but with activity, see what happens. At first, you see that the activity is giving you forces in all directions. So in some sense, it seems like it's even detrimental because it's pulling some of the membrane completely in the wrong direction. And in fact, if the force is strong enough, it can also be detrimental to the engulfment. But with not too strong a force, you see what happens. So there was a spontaneous formation of, again, of this highly curved arc, just like we had this arc on the flat surface. But on the flat surface, this arc was leading. If it was in one direction, it was giving a motion of the whole vesicle, or if it was in two places, it was simply pulling the membrane. In this case, this arc is just leading to this, like a capuchon-like engulfment of this bead. And the last part that I want you to notice here is that this arc, after it has formed spontaneously and has pushed or pulled, if you want, the membrane over the bead, it completely disintegrates spontaneously as well. So several things here are really cool, which people, when you look at the biology of phagocytosis, they talk about a lot of machinery which is supposed to be involved in recruiting the actin and making sure that the actin forms a ring, and then making sure that the actin is disassembled afterwards. And here, all of these things happen because of physics. You don't need to invoke very complicated biochemical reactions that will maintain a ring. It was not even a full ring, it was just an arc, but that was already enough. And to disperse it afterwards, again, physics is doing it for you without any complicated biochemistry. The other point that you might ask yourself is, what happens to the bead here? Can it still breathe? In the sense that, do I allow fusion here of the membrane? And I don't allow fusion of the membrane. So in fact, there is always a hole here that connects the bead to the outside world. So this membrane is topologically still a sphere, okay? So there is always still here, so if you look at the cross section, you'll have the bead, and then you'll have the membrane, and the membrane will be like this. So there's always a breathing hole for the bead, okay? In reality, of course, there are really certain proteins that cut it here in the cell, and then this bacteria is being eaten in the cell or whatever, okay? But we didn't deal with that part. Okay, now if you look at images, like electron microscopy images from cells that are engulfing special objects that were given to them, you can really see how the membrane is enveloping these beads, and you can see that it's full of actin polymerization. Actin is labeled here in red, and this is really essential for driving this process. So as I said, we can get full engulfment with the passive proteins, again, just by adding enough of them. So 0 engulfment here at low enough EAD, and just adding enough of these proteins. Well, in this case, we didn't get full engulfment, but for example, from here to here, no engulfment and then full engulfment. And you can see as function of time that you get exactly the same thing as when you got the spreading. These passive proteins, they simply line up spontaneously this highly curved edge, and they allow it to continue to move on the surface because you don't pay anymore this high energy. Now with the force, you can now get at much lower densities full engulfment just by turning on the force. So you can be at a situation where with 0 force, there is no engulfment, you turn on the force and you switch to full engulfment in a condition where the passive system, or 0 force, could not give you engulfment. So again, recruiting the actin allows you to complete the engulfment at a much lower density of these proteins at much lower adhesion energies. Then we saw this recent publication of very high resolution images of this actin ring that forms during the engulfment. And what is interesting is that because of this high resolution, for the first time they could see, they even gave it this name of teeth and jaw, because before, people with low resolution images thought that this has to be a continuous actin ring in order to complete the phagocytosis. And here they saw that no, you can have very fragmented ring of actin and it still works just as well. And that's exactly what we get in our simulation. You can see that it's highly fragmented. It's partially continuous here, but it's almost non-existent on this side in the beginning. And then as it progresses towards the end, it usually becomes more and more continuous, but it is not a condition for this process to succeed. And you can compare these experiments with these simulations. At least that's what we try to convince the reviewers. Another part which I want to show you, if I have a few more minutes, is what happens when you move on the sinusoidal surface. So we found this interesting paper from Korea from 2015, which is again in Biomaterials Journal. So maybe many biologists that we talked to didn't hear about this paper even, although they work on cell migration, and it's easy to miss papers that are in more engineering journals. But what they found is that when they put these cells on these grooves on this sinusoidal surface, they tend to move only inside the grooves and avoid the ridges. And if they do cross from one groove to the other, they tend to cross at a very high angle, so almost perpendicular to the ridge, as if to minimize the time that they're on the ridge. And then again, they move on the groove with rather happily inside the groove. And when we tried to simulate that, we found very similar behavior. When we started with our small vesicle already in this polarized state, so already perfectly motile on the flat surface, that was our initial condition to make life simple, and we let it go perfectly aligned along the groove in the beginning. It seems to continue to go in the groove. This is the trajectory. It is losing its polarity over time. You can see that its speed is going down. That's in the color. But it doesn't go out. The ridges are shown by the red lines. That's the peaks of the ridges. It doesn't try to go up. When we put it on the top of the ridge, but again, aligned with the ridge, you can see that it very quickly slides off the ridge. It's not because of any gravity, of course. And it slides off the ridge at a very high angle, so almost perpendicular to the ridge, and then settles again inside the groove, very similar to what they saw. So when we saw that, we said, great, we can explain these experiments that nobody explained from 2015. I can show you the movies. So that's the experiment, and you see that it slides off, and that means that indeed you move almost at right angles when you cross between ridges. So you can see in the trajectories that crossing between ridges is almost like a 90-degree turn in the trajectory. And when you go in the grooves, you see those parts of the trajectories which are very long, relatively longer and straight, and that's how it looks in our case. Now, it loses velocity in our case because, as you remember, our vesicle is very fragile, and it is perfectly moving on the flat surface. But here it's encountering these boundaries, these curved boundaries, and that means that from time to time some of the proteins break away from this large aggregate. And where do they go? Well, they begin to form their own small aggregates in all kinds of places, and that means that the net force in the forward direction is reducing over time. So, as I said, we have limitations with how long we can run these simulations because eventually our polarized object is fragile. We don't have these internal mechanisms of the cell that maintain polarity. Now, you can ask why. What is the reason that your vesicle doesn't like to be on the top part of the ridge and likes to be on the bottom? You have a simulation. You can interrogate the simulation and ask everything. You have all the energies. You have bending energy. You have pushing energy. I mean, you have so few energy terms. You must get the answer. So in order to do that, we thought maybe we can go to an even simpler geometry. I mean, on the one hand, people did the sinusoidal surface because it sounds simple, and it is simple, and it's also easy to visualize because all visualization in biology is almost done on a 2D system because of optics. But if you think about it, the curvature that you are encountering is constantly changing from positive to negative, positive to negative. It's difficult to understand what goes on, especially if your vesicle can be sometimes part of it on the positive curvature, part of it on the negative curvature. So why not put it on a constant curvature system, like a cylinder? And we did. So again, what Raj did is that he took this perfectly polarized object, he mapped it onto the surface of the cylinder, perfectly aligned along the cylindrical axis at time 0, and let it go. Okay, and see what happens. It starts off going straight, but very quickly, it doesn't want to go straight anymore, and it prefers to start rotating. And then it can rotate on and on and on like that forever. So that is the reason why on the ridge it wanted to go at 90 degrees, because it thinks it's like on a part of a cylinder. And apparently on a cylinder, it wants to rotate perpendicularly. And that's what it was thinking it was trying to do on the part of the ridge. And now you can ask, okay, but why? Why does it want to rotate and not go straight? What's the difference? So as I said, we have all the energy terms, so we can see what happens to the energy when it starts in this state as compared to when it is perfectly rotating. And you can see that the adhesion energy is slightly getting better, so it's decreasing. The bending energy is slightly getting better, but again, look at the absolute values of these energies and look at the changes. The changes are very small. And there's also the binding energy between the proteins, which is slightly improving, but that's already now in single digits. This is a few tens. This is like, I don't know, like 8 or something like that. So very small amount of energy difference. And we think that what is really happening here is that if you are oriented along the axis, and you are trying to spread as much as possible. So what this arc is trying to do is trying to spread along the surface to increase the adhesion energy as much as possible. Then the pushing force forward is effective because it moves you on the membrane. But all the forces on the side, they're already pointing in some sense outside of the cylinder, because you have something which is like that. And all the forces which are here, they are pointing away from the surface. So they are not they are not as effective in spreading you on the surface. They want to spread you on the surface, but it's a highly curved surface. So spreading in this direction is more difficult. On the other hand, if you're oriented along the circumference of the cylinder, this direction is pulling you, but that is already, you know, the fixed size of the arc. But the sides, which are the most effective in stretching the membrane and spreading it, they are much more effective now because they are spreading this whole shape sideways along a zero curvature direction, which is much more effective. So that's why we think this is much more effective in spreading the shape sideways as opposed to in this direction along a cylinder. So you are able to slightly stretch this whole object more in this configuration, and stretching it and increasing the contact area is identical to increasing the adhesion energy. And you do that without costing bending energy, so you're also slightly improving your bending energy. So that's our understanding of what goes on here. Just to show you that you can actually, you can actually start with a two-arc object on the same cylinder, and now each of the arcs by themselves is trying to rotate, but when each of these arcs is trying to rotate, it ends up coiling your two-arc shape. And this explains maybe naturally why membranes, when they have this elongated structure of like a protrusion which has a leading edge which is pulling it on the fiber, it cannot actually stably move straight, and it spontaneously begins to spiral. And we have examples of that from these cells which are doing myelination. So these are cells which are glia cells which catch axons, and when they catch the axons, they spread on them, and they begin to spread this myelin sheet on the axons, which allow the efficient transmission of the action potential. If you look at the shapes, they really do look like spirals. And it's funny because when we approached the lab of Ori in Weizmann, one of his main research assistants told me that when she's doing this research on the myelination process, they are usually not interested in these initial stages of how the glial is first spreading itself on the axon, because that happens very quickly, and then the myelination process itself takes hours to days later, and that's what they are really studying. But she remembers that when she just looked at the initial stages, it looked like a spiral, but nobody ever reported it because nobody cares. And because they're only deeply interested in the myelination process, which happens much later. But the spreading is, of course, essential in order to myelinate, but they took it for granted that somehow it spreads. But you can see that she was right, and when she went back to do new experiments, she did find these spiral protrusions. So, I think, we are really excited by this idea, which, as you saw, started long time ago with this work with AJ Gopinathan, on this coupling between the curvature of proteins and the active forces they can produce. So far in the simulations, we had only isotropic curved proteins, and already it's very rich. But as I already said several times, it seems that in reality, they are highly anisotropic. Even a simple banana-shaped protein is highly anisotropic. And we really want to expand this research in the coming years. This is something we are very much excited about. It can give us maybe explanations about motile cells and many other processes in biology. We want to add anisotropic proteins, as I said, mixtures of proteins of different curvatures, which already I showed you yesterday at the linear stability analysis level can give rise to propagating waves. Contractile forces, which again I showed you yesterday, can give rise to propagating waves, and effects of filament bundling, because so far we said that the local outward force is towards the local outward normal, and that seems to be reasonable for branched actin network where it's like just isotropic pressure. But if you have a very strong bundling of actin, so the force, you cannot say anymore that the force is normal in all directions here. It's really only along this direction, because all the actin which is polymerizing is really only giving you a force in this direction. So somehow to think about, since we don't describe this explicitly, somehow to think about these proteins as not only giving force in their normal direction, but somehow aligning themselves both to give a force which is in the same direction, because they are close to each other. So somehow to think of something which is a little bit like the Vicek interaction of alignment of flux or self-propelled particles, but now on the surface of a flexible membrane. So it's a new idea, I think, also in terms of flocking and self-propelled particles. Yeah, so I thank Raj and all the guys in Ljubljana, and soon I guess I'll also thank Shubhadeep, who is the new postdoc, and thank you. Yeah, okay.
I have a sort of a general question. Yes. So these protrusions, so this is related not just today's lecture, but also yesterday's. These protrusions that come out, so this is sort of a selection problem where it comes out and how it happens, right? So if I look at similar problems like Sussman-Taylor fingering, that's also an instability that gives rise to a finger, which is somewhat like these protrusions. So can one say that this effective force that you are putting in here to capture the active force is analogous to some force that arises in the Sussman-Taylor from the interfacial tension problem? I mean, is there something that is similar in the two problems?
You can try to think of many analogies in other pattern formation systems which are out of equilibrium. For example, a system where you have two fluids of different densities in gravitation, and the higher density is going into the lower density, and it forms these protruding fingers, which then have these eddies forming on them. So in the case of the gravity, the external source of energy is gravity, and the fact that you started off with a high density above the low density, which is not the minimum configuration, minimum energy configuration of the system, gives you this
driving force,
this slope on which you can go and produce the forces. So in some sense, each of these active forces is represented in the model as an energy slope, which is like an external field. And this external field can be thought of as gravity in other systems, or an external electric field, if you want. And you need to pump energy into the system.
Yeah. Another related question is that if I look at similar one in the case of dendrite information, the front propagation there. So there, of course, the branches also appear. But I didn't see any branches in the patterns that you looked at. Yeah. So what prevents branching?
So you didn't see branching here, and there's several things that can prevent branching. First of all, this system, I don't know that it should also show branching, but a similar system does show branching. What do I mean? In this case, we had so in this case we had that wherever you have high curvature, you also have an aggregation of these proteins, right? And therefore you have a large force. So the force increases with the local curvature through the increase in the aggregation of these proteins. But if you put now the proteins out of the equation and you forget about them, and you simply say, I have a contour, I have a membrane that by some reason is active in the way that wherever it has this kind of curvature, it has an outward force which is increasing linearly with this curvature. So when it's flat, there is almost zero force, and when it's negative, also zero force. So you think of some force versus curvature that has something linear like that, only in this positive, or if you want, a negative curvature direction, it has a force which increases linearly. This system we have solved, and that's very similar to the Sikerka instability of dendritic crystal growth. So what happens here, if you use this simple rule, you say, okay, forget about these proteins. Simply what you say is that whenever you have high curvature, you have a lot of these proteins and the force is loud. So let me just simplify and work only with the membrane itself, or impose on the membrane this simple rule. Then you find that as this thing is growing, it undergoes tip splitting. And this tip splitting can undergo another tip splitting. And this does give rise to this dendritic tree that you are talking about. And we suggested this simple rule for the progression of an edge of a multicellular monolayer, where you do this scratch assay and you look at the wound healing experiments. And on these monolayers, it was found that there is the formation of a leader cell, and this leader cell made these lines of many cells moving together grow. And it seems like on some occasions you could find that a single leader cell didn't split physically, but became this one, and another one was forming on the side of it, and you could really follow this tip splitting. But even there, it was very rare in reality, not in the model. And the reason is that in reality, this is not a continuous contour, but it is discretized by cells. And as soon as you even run a model which is discretized and not anymore a continuum model, you find that just by the discretization, you've suppressed very much these tip splitting events. There is still the tendency to form tip splitting, but because everything is highly discretized now, it is very much suppressed. So that's what I can tell you about dendritic growth. But in this model, which has these proteins which aggregate and they produce the force, which is more complicated than simply imposing such a rule on the membrane, so far we didn't see any dendritic growth, and I don't know how to, I don't have any mathematical result that shows me that I should get. Here I know that I should get. I can show that it becomes unstable.
Okay, thanks.
So yeah, so you're right that in all these simulations, I have only blue, which is no proteins, and red, which is one kind of proteins. And you're rightly saying that in the cell, there are many kinds of these curved proteins, all existing at the same time. So what goes on? So thankfully, I think in the cell, not all of them are recruited to the same place at the same time. How that is done, it's a very good question, but that's beyond what a simple physics model can deal with. It has to do with the biochemical control of all the cells, and there's lots of biochemical cycles that hydrolyze or dehydrolyze, phosphorylate or dephosphorylate a protein, activate it or deactivate it. Only in the active form it binds the membrane, in the deactive form it's just not on the membrane, so you can forget about it for the membrane shape analysis. So many of these curved proteins exist, but not all of them are recruited to the membrane at the same time and at the same place. But you're right, surely there's more than one in these protrusions, and many of them are redundant in the sense that you can knock out one, but still you get the same protrusions, more or less. So you can say, why should it be there at all? But as was mentioned in one of the talks, you cannot understand life without evolution. So, why was this redundancy formed at some point in the evolution? It's not a physics question. But in principle, you're right. You have many of these curved proteins, and they will tend to aggregate, if on the membrane, in places that fit their shape. And therefore, they should follow behavior or dynamics, which is similar to what we are showing. Yeah. Yeah, but this is when there is active forces. You remember that when these curved proteins had no active forces, and I think caveoli is not recruiting actin, then they form just nice spherical buds. So very much like caveoli. So on that. Yeah. Yeah. Yes. Yes, I expect so.
Yeah, hi. In these bottom left simulations, what would have happened if the structure was already in the most, like the least energy state? Would it still have this kind of rotation around the cylinder?
Yeah, if you see this crescent shape has all of the proteins on one side. So
Right.
if you sum up all of the forces, they tend to
Have
keep
a
moving in the same
direction.
direction. And in fact, they must do that because if they don't push, then you lose the high curvature of this rim, and then the highly curved proteins will go away from there. So the force is sustaining the highly curved shape, which is sustaining the aggregation of the proteins, which give you the force. So this is
And
exactly the
in
positive
the
feedback.
two head cases, the forces are in the opposite direction, like the top right.
Yeah, but then they're on opposite directions, but
Yeah.
they
But
are.
so when they come together, they stop rotating. Is that the reason, or?
Well, first of all, they keep on stretching the membrane, so the membrane keeps on being highly curved there because the force is still pushing on the nodes. But first of all, we don't allow them, of course, the membranes to do anything interesting when they interact
Okay,
sideways.
I see.
They just avoid each other, so there's nothing interesting happening there. But you basically run out of membrane, so they coil and then they are stretched, and then that's it essentially.
Right. And I maybe missed this, but what is the interaction potential between the membrane and this surface?
Oh, it's always the same. It's simple adhesion.
Simple adhesion.
Adhesion.
Can you just tell me the equation, or like
Yeah, it's just
LJ
a
or
step
something?
function. It just means
Just a
that,
step function.
yeah.
Okay. Okay. Thank you.
I have a
Yeah, so I have a question. This seems the net force on this movie, this net force is acting only in a certain polar, I mean certain direction, and the coming out for these car proteins. I mean, if you look at the movie, it's going into in one direction only.
So what you mean here?
Yeah.
Yeah, but that's because they are organized in one direction. The local force from each of these red dots on the membrane is directed towards the outwards normal. It doesn't know what the others are doing.
I see. So
there's no communication between them. It's completely local.
I see. So, and it's always generating unidirectional motion in that sense. That's why they are going in one direction.
No, you see, this is not unidirectional. And there, after the engulfment finishes and before it starts, there's nothing unidirectional. They don't know about each other. It's only when they self-organize into some structure that there is a net force in a particular direction. But before they self-organize into some structure, there's nothing to tell them where to aim. The force is always towards the local normal.
No, so
outwards normal.
So, I mean, is it like one of those configuration that you see these, or it does not go other way? I mean, is there, how does it pick up only these?
You mean why does it rotate right and instead of left?
And also,
that's random.
huh?
That's random. That's random. Yeah.
Okay, that's
And you mean why does it go to the left initially? That's the initial conditions. You're free to start any initial conditions you want. No, it doesn't matter if we start it to the right or to the left. You agree with that? So, but does it choose to rotate left or right? This is done randomly.
Yeah. So in your models where you had initially protein without proteins, you were explaining the curvature. Then you had proteins without activity, and then with activity. So, like you were explaining it in terms of energy, but I had this intuition. Like, can we explain this in terms of the balance of forces? For example, when there's no activity, we can assume that there are random forces acting which are not allowing the proteins to cluster at one place, but when there is activity, they are clustering together because of their activity, I mean because of their forces on the membrane.
You mean when they are on the rim of this spreading object?
Yeah, I mean when they are actually, when you start the simulation and throughout the progression of your simulation.
Let's go back there 1 second and we'll quickly understand the question better. 1 second. Okay, I'm almost there. So we're talking about the spreading, which is before any of this motion.
Yeah, in these three cases.
In these three cases, for example, yes. So we have the case without any proteins. So basically you have only the balance of adhesion and bending energies. That's only the energies that you have. Then you have the passive proteins. So you have also some aggregation between them, but you see that the aggregation is not very strong by itself because they are highly dispersed. But as the spreading is progressing, there is a more and more highly curved rim which is forming, and that tends to aggregate them more and more along this highly curved rim because the bending energy is reduced when they go there. And that helps the spreading even more. So there's a positive feedback between their aggregation along the rim and the continuing spreading until, in this case, it's almost fully spread. Okay? So, but again, there's no forces here. It's only energies. Or well, maybe I'm missing what you're suggesting.
No, like irrespective of adhesion, I was asking irrespective of adhesion, like the clustering of proteins happening on the membrane, can it be explained in terms of forces? Like the forces that proteins are inducing for inducing the curvature on the membrane and coming on the rim, and the forces which are opposing that.
But without adhesion, the passive proteins don't form this rim.
It's fair only when you only
They
have
form only
stress.
budded objects.
Yeah.
But why would it be easier for you to think in terms of forces? Usually thinking in terms of forces is more difficult because thinking in terms of forces means that you have to solve equations of motion. You have to solve dynamics. And that's always more complicated than doing thermodynamics because in the thermodynamics, you only deal with the free energy and you try to find the configuration of minimum energy. It's usually much, much simpler than solving equations of motion. For example, you saw that the Helfish Hamiltonian is very simple. It's just a simple quadratic form of the curvature. But when you want to convert it into equations of motion of a membrane, you get these force derivatives and all of that. So it gives you equations of motion which are much more complicated to integrate in time than just minimizing shapes. Yeah, it's easier to minimize shapes in this case.
Sorry to interrupt this discussion. Maybe we should break.
Yeah. Okay.
Reassemble at 11:15, I guess.
You're welcome to write me emails with questions and suggestions, and we can continue the discussion. And also, if you're interested in postdocs, you can ask me about that and other things. Okay, thank you.